So in the last section, we've been able to do step-by-step approach in exploring comprehensive and functional genomics. So we've been able to assess WGS reads on NCBI database. And then we use European Nuclear Archive to view samples unique to the accession number that we copied from NCBI SRA database. And the use of Patrick database to do reads processing and genome assembly. So the next video, we explore genome annotation. We submit the SRA assembled data set, the assembled FASTA Contig, right, to, Patrick database for genome annotation, all right, and then gene prediction. So we'll be evaluating the results that, of the analysis we have done so far, which include the result for genome assembly and then gene prediction. So in evaluating the results, we'll be looking at reports analysis of some submitted sample for genome annotation, which included the one we used, right? And reports for genome assembly, report for genome features, report for protein features, report for specialty genes, report for pathways, and then we're looking at visualization of genome functional features on Genius software. So here on Patrick, we want to explore the genome annotation that was done for this particular organism in the last video session. So I'm going to view it right now, as you can see. All right, so the result is loading. Now, as you can see, here are the results of the genome annotation that was done from the service area, for this particular organism, this mycorrhizal fungi that we, submitted. So right now, we have, the mycorrhizal fungi, the accession number, the Contig.Fatsa. So this is the Contig.Fatsa file that we actually submitted. And this is EMBL, is another kind of, file format for these results. But we don't need that right now. And this is for protein. So this is like protein file, okay? The protein file, you can download this, the DNA, file, but the futures, meaning DNA futures, which is the CDS, and this particular protein features contain, the proteins like, the functional proteins that were annotated for this particular, microorganism, okay? But this particular mycorrhizal fungi, all right? So we have this, and then we have this. So this is for DNA, feature, which is the genomic feature, while this is for the proteomic feature, since, it is protein, all right? Then we have the futures.txt. So the text file that contains all the features of this particular, genome that we submitted. And this is GB, which is gene bank. So this gene bank contain, both the DNA and the, protein features. And then we have, the genome. Then we have this March GB. So the March GB is, the merge file, the complete genome match file of this particular genome annotation that was done. All right. So, at the Contig.Fatsa file that we submitted for genome assembly, right? The result we get for genome assembly, which is the Contig.Fatsa. We have the contigs, and so many of them were, segmented like in fragments. So the sequences in the file of the genome, right, is in fragments. That's what I mean. So, but here it's no longer in fragmented, right? But all contigs has been merged as a whole genome. So, and then this is the text file of, the organisms, probably miscellaneous features, okay? And this is the genome reports.html file for the genome annotation that was done. So we're going to start from here, and then we move further to downloading some of these reports, and then we explore them. But meanwhile, let's try to explore this so that you have a better understanding of what we're about to do. All right. So I just clicked here, and I'm going to view from here. All right. So as you can see here, we have the genome ID, which is the ID for this particular genome annotations that we did. Now here we have genome name, which is this actually plus the session number, the reference genome norm. The cost consistence and file consistence is the function of the quality of the annotation that we did, the quality of annotation. So cost consistent, file consistent is good. Like cost consistent, we have 93, and file consistent, we have 92.8. This shows that the annotation that was done was highly processed. Okay, it was highly processed, was was annotated, and the annotation is in a high good condition. Okay, so it means the prediction of the genome features and the protein features, which is genes, and there's some enzymes, some proteins, and everything, the entire features that is found in the genome sequence, right? So the prediction of those genome features is of high process rates. So meaning that 93, it means like the prediction was like 93% done, right? Like the prediction, the rate, the level of the annotation done that was gotten was like 93% out of 100. So that is in high condition. All right. Then we have completeness to be 4.2, which is okay. Then we have contamination to be 0. So we have 0 contamination. Completion is this. So we have like maybe 42.4.2 yet to be complete. All right. So we have Contig count. So we have like 791 Contig that is found in this genome. And then we have evaluation to be root. So the root is like the taxon name, and the taxon name is this, right? Of this, that is the taxonomy name of this genome that we are of this particular organism. Okay, genome. Then we have DNA size to be this, Contig.N50 to be this. So the entire DNA size is this. All right, that is like 4.6 million nucleotide bases that is found in this particular genome. So that's what it means. DNA size of the entire genome is this. So 4.6 million plus nucleotide bases that is found in them, the ACGTs. Then we have the Contig.N50, Contig.L50. So the Contig.N50, which is the base pair, right? That means in number, where L50 is in length. So the N50 is like the numbers, right? In numbers, which is the base pair numbers, and the L50 is this. Okay, that's the length of the Contigs. And then we have overrepresented rows, which is 17. Underrepresented rows to be like 81. So this is just talking about the rows that were predicted and functionally presented in the genome. Then we have predicted rows to be this. So this is the entire predicted rows that we have, and then completeness row to be 24, total distinct row to be 100. Protein encoding genes with function assignment, we have 133. Protein encoding genes without function assignment is 10,407. So those without function assignment are like apothetical proteins, and function assignment is 135. Now this is just the extent of this analysis that was done on this particular mycorrhizal fungi, right? So if we are to explore the same thing for other organisms like bacteria, which you can try, okay, just get the SRI accession number and try. You get to understand that the functional assignment will be more, while the encoding protein genes with functional assignment will be more, while the encoding protein without functional assignment will be less. Why? Because this particular database is curated to mostly annotate bacteria. All right. So here we have protein encoding feature coverage to be this, and then features that are apothetical to be this, features that are in local proteins, to that's a value. All right. So, but this is just you don't have to worry about this. We still explore more. So this is just the overview of the genome annotation that was done for this particular genome. So if you are given a genome sequence for you to annotate, all right, or you are given any numbers, so when making reports, this is what you actually need. Okay, these are some of the things you can actually write. So if you are working with maybe 4 to 5 samples, you just have to form an Excel table where you'll be able to present these numbers and be able to report that as your main report of the work you did. All right, so now let's try to explore the genome itself. All right, so I'm going to click here this number, and once I click this number, it will take me to a full detailed report where I'll be able to narrow down my research to specific genes according to what the basics of functional annotations is. Genome annotation is about. So I'm going to click the genome ID. So it takes me to another tab. So it's loading. It's loading. Now, before that is completely loaded, I'm going to show you how you can come to whatever we're about to see here, all right, which is loading already. So I'm going to show you how you can get to this particular point without clicking this number. So let's just go back. All right, so you can actually click this eye icon here. So the moment you click this eye icon, you'll definitely be automatically redirected to this point. So this is for the number, okay, and this is our this is the taxonomy classification of this particular fungi, right? And this is our own sample, and this is the result. As you can see, the genome features, the apothetical proteins, and I mean the protein features, including the apothetical proteins, protein with functional assignments and others. And then we have specialty genes to be here. So that is the specialty genes. And then most importantly, let's try to see the result. Let's see, can you see it's the same? The report from clicking the eye icon, and this is the report from clicking the genome ID. All right, so all the same. So let's just go back to the previous here. So we will come back to exploring this deep result. Probably let me explain this. Then we go back to explaining all of this. Try to explore this single individual report. So we saw the overview of the genome annotation that was done, including the cost consistence, 5 consistence, talking about the numbers of apothetical proteins and encoding genes with functional assignment. So here, as you can see, we have genome features, the CDS, the tRNA, and here we have the apothetical proteins with functional assignment, go assignments, and then we have others. So included in this result, we also have the specialty genes, which includes the virulence factor, the transporter, the drug target. Now, all these are genes, all right? All these are genes, right? They are genes. They are protein features, right? Some of them, the protein features has been classified, like the functional proteins, have been the functionality of those proteins has been classified, while some they are yet to be classified. Now, how do we explore this CDS, right? Now, what is CDS? CDS is coding sequence, meaning sequence that actually codes. Now, these are the exons, all right? The exons are regions that can actually code for proteins, and these coding sequence are exons, like areas of proteins, CDS, like area of DNA sequences that actually code for a protein. So that is what we call CDS, and we have a total of 10,552 coding sequence. All right, 10,552 coding sequence, like potential proteins with functional assignments. All right, so but how do we explore this CDS? And then we have tRNA also to be 7. All right, how do we explore that? Now we just have to go to futures, because these are genome futures. So in doing this, just go to futures. And in going to futures, this is where you are, you'll be able to do some gene predictions. Now, don't forget, when we submit genome annotation, genes have been predicted, but then to be able to narrow down our research to certain specific gene, all right, of interest in our mind, all right, we just have to come to these genome futures where we'll be able to filter, all right, we'll be able to filter, or we'll be able to search based on keywords. We'll be able to search based on keywords. So if you're exploring a fungi, you probably are exploring a bacteria genome, all right, and then in your research, you have a specific gene of interest in mind that you want to search between these microorganisms or between these in the fungi, all right. Or maybe you have 4-3, and then you want to look at differences that occur at certain specific gene that is found in each, in different 5 organisms, all right, or microorganisms, be bacteria, or when you're exploring fungi, all right. So what you just have to do is you use keywords to narrow down your research to that specific gene from the CDS, because the CDS includes those proteins that are hypothetical, right? When I mean hypothetical, proteins that their functions are yet to be predicted, and including proteins that have an encoding gene whose functionality rather has been predicted, all right, has been assigned. The functional role has been assigned. So you can search based on this. So as you can see from the overview, we have that hypothetical proteins is much more than the functional assignment, all right. It's much more than functional assignment. So it means we have less functional proteins, or proteins whose functional role has been assigned. All right. So, but as we explore, can see we have more hypotheticals. We have more hypotheticals. So you can filter based on CDS and tRNA. So you can remove this CDS and just try to explore the tRNA. All right. You can choose to explore the tRNA. So I just click the tRNA now, and it's loading. It's loading, and we just have 7 results, and these are 7 results. Can you see these are the tRNA products that we have? You can choose to view more functions here, more features here. Just try to see what you have, the genome ID also, and then we have the go terms, and some of the features that you can actually explore here. All right, and then you can choose to remove the tRNA and try to explore the CDS. All right, you can choose to explore the CDS. Let's just try to go back again. All right, so we are back, and then let me try to see if we'll be able to explore a bacteria genome. I already have a bacteria genome, and as you can see, this is the bacteria genome I got from NCBI database. I submitted is Lactobacillus fermentis I4. This is another. It is Lactobacillus is a bacteria, and this is the strain. It's a specific strain of Lactobacillus fermentum, right? So as you can see, we have more genome features, which is CDS report repeat region. We have tRNA, CRISPR repeat rRNA. Now these. And then we have CRISPR array. All right. So these are genome features, right? Encoded, genome features, encoded genome features with functions. All right. So and then these, as you can see, the hypothetical proteins here is lesser than proteins with functional assignments, right? Hypothetical proteins are actually proteins, and proteins functional assignment also proteins. But like I said, these proteins, their functional role has been assigned. All right. So this is bacteria. We tend to have more results here, and the same also the gene features. So the reason why I'm showing you this is because we have more gene features in case when you are exploring something else and you're not just seeing CDS and tRNA alone, you are seeing rRNA. As you can see here, you're also seeing CRISPR, and then an other kind of and repeat regions, as you can see. So these are the genome features that we have, and then we have go terms, which is geo annotations, and we have the pathway assignments, which we are also going to explore. Then we have the specialty genes, which is transporter, drug target, and antibiotic resistance, and these are the value. Now this value contains the numbers of genes or proteins that are found in each categories of feature. All right, in each category of features, you can see we have genome feature here, and this is the total value of CDS that is found by that particular genome category, genome feature, right? And here we have protein feature here. Right now we have hypothetical proteins, and this is the future. I mean, this is the value of this particular, that contains like, the proteins, the total number of proteins that is contained in them. Now let's go to futures, and right now we have the CDS and the Patrick. I mean, the annotation types we have here is Patrick. You can choose to remove this and just focus on the CDS, and then let's see what we have. Yeah, it's completely loaded. As you can see, we have less hypothetical proteins and more functional, genome features, as you can see here. So all these are genes, all right? All these are genes. We have some genes who have EC numbers, which is enzyme commission. Can you see enzyme commission? Meaning that this particular protein here is an enzyme, right? It's an enzyme. And it has a specific function that is, being a particular function that is being enrolled, right, that is functioning, right, that is encoded, right, a particular function that is encoded in this particular bacteria, all right. So we have here to be a place where we can search the keywords, all right. So what are some of the genes of interest that we could, let's look at biotin. What is biotin? All right. So biotin is a B vitamin found in food, right? Found in food. And this particular bacteria here that we're exploring is a probiotic, right? It's a probiotic organism, right? So it means this particular organism can be used as food in the food industry, all right? So, but biotin is like, it's a vitamin, all right? So let's just search biotin, all right? Let's search biotin from these keywords. We want to narrow down our research from this 2,170 something. And can you see? So it means we have like 6 biotins, right? 6 biotin of 6 potential vitamins in this particular organism. 6 potential vitamins, all right? Because biotins are B vitamin found in this. So we have like 6 category of B vitamin, which is biotin, that is found in this particular organism. As you can see, I narrowed down my research from about 2,000 plus genes to just this one, all right? So you could narrow down your interest, right? Your gene of interest based on anything you have in mind, all right? Based on your desire, your research, what you're doing. All right, we have what we call mobile gene elements, And okay, let's just try to search what is mobile gene elements. All right, so mobile gene elements is a segment of DNA that encode enzymes and other proteins that mediate the movement of DNA within genomes or between bacteria cells. All right, so that is the function of mobile gene elements, and it's a protein, right? They have functional roles. So, but what are different types of mobile gene elements that we have? All right. What are mobile gene element example? Can you see we have plasmid, antibiotics, we have CRISPR, so which means the CRISPR we were viewing, we are looking at from which is these are also mobile gene elements, all right? The CRISPR you see here, also mobile gene element. Let's go back to the futures. All right, so we're back to the futures. So we have like mutations, we have so many of them, and other kinds of mobile gene elements that we have is we have transposase, transposomes, and things like that. All right. Transposase, let's look at Wikipedia. What did Wikipedia says about mobile gene elements? And what are some of the examples of mobile gene elements we have? So we have is a genetic material that move around within a genome or that can be transferred from one species to another. So this is just mobile gene elements. And some of this mobile gene element carries certain things, right, that could affect other genomes in as much that has been transferred. So some mobile gene element carries some features, right? So and once it has been transferred to another, can you see from here, to another bacteria or another organism, right? It could affect that organism, all right? It could affect it because there's a transfer, all right? There's a transfer. Now, this example, can you see the types? We have transposons, we have interons, we have DNA transposons. We also have transposase. Can you see CRISPR? So can you see interons, retrotransposomes? So we have them like that. And then let's just try to copy these transposomes and let us search from the CDS, and then let's see, do we have results as mobile, a mobile gene element transposon? All right, so let's remove the S and let's search. Okay, we have others to be transposes. Let's see, do we have transposes? All right, we have transposes, which is 60. So we have like 60 of transposes, right? Transposes, like 60 of them. Let's see, let's try to check the back. What's that? Integrons. I see even RNA, introns is also there. So let's see. Do we have integrons? Okay, no, we don't have integrons in this particular genome. What about introns? I don't think we have introns also. Let's try it again. All right, so it's not present, okay? It's not present in it. So, but this is how you narrow down your, from here you narrow down your gene of interest, all right, to certain specific, region, all right, that of interest, all right. So this is how you do it. You do it from searching for it exactly from here. So probably you could have, certain genes, abbreviated genes, okay? You could have certain abbreviated genes in mind. So this is how you also view that, all right? You also view that from there as you're shown. Let's see. So sometimes mobile gene element can also be called NG, all right? Can also be called NG. So this is how you could, let's say, glycolysis. Okay, what about genes on glycolysis? How about genes on glycolysis? All right, I think let's check it this way. Let's check glucose. All right, we have glucose. We have genes on glucose. All right. So we have different genes of interest you could have in mind to to narrow down your research, all right, depending on which functional gene element you have in mind, in particular to the organisms you're studying, be it fungi or bacteria or even human. If you are annotating human human sample, or using another kind of database, right? Another kind of database in mind. All right, so that is just for the genome features. All right, so and then we have the functional assignment, which is also included in the CDS. All right, and then we have pathway. So the next thing we have is pathway. But then let's see. We've explored the last time we explored genome assembly. All right, in the last video, and then we have a report on genome features, protein features, specialty genes, and their values which you have shown. Okay, let's look at reports on pathway. Let's look at reports on pathway. All right, so let's go to pathway, and then from pathway we have the EC numbers, which is enzyme commission numbers, and then we have genes. So, but the pathway is loading. So we have different pathways. All right, we have different functional pathways. So we have like metabolism, carbohydrate, stuff like that. So you can filter, and as you can see, we have carbohydrate metabolism, amino acid metabolism, biosynthesis of secondary metabolites, xenobiotics. So you can also choose to narrow down your research depending on which particular pathway, functional protein are you looking for. All right, pathway functional protein. Now, the reason why we have the word pathway here is that for every functional features that is running or processing in an individual organism, all right, all those functional proteins as pathways, right? They have pathways at which they are functioning, they have been processed, all right, at which they move, at which they function, all right, at which they operate, all right. So now those pathways, we have them as in classes, all right. We have the pathway classes here. So each pathway classes has the numbers of genes present in them. So do you have a particular gene of interest based on a functional pathway classification, right, in mind? Probably your gene of interest is to explore the area of carbohydrate in an organism. So what you just need to basically do is to go to the pathway where you'll be able to, which is another area of functional classification, and you just click your carbohydrate metabolism from the filter here, and then you click the filter back. And let's see, loading pathways. This may take several minutes, but let's exercise patient as it loads. All right, before that is done, now when you are constructing your results, all right, you can choose to create an Excel form for some of these reports, and then you can report in words, right? You can import in words that this particular genome has the value for, the CDS, tRNA has this value, the political proteins have this value. You can also form a table, like I said, and then, give some, words. And if a particular client, or maybe you as a researcher, you have something in mind, a gene of interest in mind, just like I said, just go to features and narrow down your interest to that particular gene. And next thing is, how many categories of that gene do you have in the genome, all right? So you also list that out. And then most importantly is, how do you download those genome features, all right? You also need to know how you can download each specific protein genome features, all right, from the entire genome. So you don't just see the numbers, the value alone. And when you go to features, you narrow down your research. How do you download so before this is done, let's go to features, and let me show you how you can download just a sequence for a particular protein, function, or gene. So let's still look at the biotin. All right, so let's set this particular one, and you want to download the sequence. So just go to click FASTA here, and if it is view protein, FASTA protein, you can click that. If it's DNA, you can click that, which is the nucleotide basis. All right. All right, can you see? This is the genome DNA nucleotide basis of this particular biotin protein, all right, biotin protein, or gene, right? So you can download from here. And the moment you download that, you go to your downloads and open it, just like I did, and here it is. All right. So you can choose to import that into Genome software. You can choose to import that to Genome software. All right. So let's try to create a new folder for that. So let's just use that, and then let's upload, import from file. So at the download, all right, this is it. Can you see biotin? And this is, can you see, this is the this is the sequence. Those genes you're interested. So probably you are interested in just one particular gene in the whole genome. And then you have a genome. So once you did annotation, you narrow down your research to that specific gene, you download the gene, you get a sequence. All right. And when you get a sequence, you can do your further analysis based on whatever you want to do, but be it variant analysis, trying to understand mutation that might exist within this particular genome, that a particular chemical compound was applied to, and a control. So you get this particular gene, once you did annotation for the control and you did annotation for the one you applied chemical or a compound to, all right, try to see the effect of the chemical compound drug or whatever you apply to that particular organism. And then you try to see what is, based on variant analysis, but that's just another analysis. That's just another research on its own. So you can do that kind of analysis, and then once you understand the variant, you can understand effect prediction on protein, all right, on the protein function. Okay, what protein is affected and what is the possible effects that it can render on that particular organism. So that's just the aspect of variant sequence analysis or mutagenesis, study. All right, so let's go. Right, let's go back to pathways. No result yet in pathways. So meanwhile, that is just how you can download. All right, so this is the pathways. We are trying to look at carbohydrate metabolism. All right, so we have the results. Can you see? So we have the result here, carbohydrate metabolism. And under that, we have glycolysis. All right, we have glycolysis. In that case, let's try. Searching for that same glycolysis again. Still, but we have it at the pathway, all right? Glycolysis and gluconeogenesis. We have citrate cycle, pentose, phosphate, fructose, maltose metabolism, galactose metabolism, starch and sucrose metabolism, amino sugar nucleotide sugar metabolism, pyruvate metabolism. So these are, because we're actually viewing, carbohydrate metabolism. So different categories of carbohydrate metabolisms are what are, those are what, is actually listed here. All right. So you could choose to explore one of it, and in exploring one of it, you can just try to still click, try to probably view the DNA sequences of just glycolysis pathway there. And in doing so, you'll be able to view the sequences. So you may have different genes also here. As you can see, we have several genes. All these are different genes of, glycolysis, gluconeogenesis pathway, all right, under carbohydrate metabolism. As you can see, so you can choose to download this, and then probably you focus on which of the gene you are interested in, or you can download this for your, if you're interested in glycolysis pathway under the carbohydrate, metabolism pathway, all right, category. So you have your control sample, you have your, your other sample. You just have to do this and try to understand mutations that could have, emerged, in your sample, compared to the control. All right. So this is spectacular in a way. So now we could choose to explore only the genes, try to look at the genes one after the other. So I just click genes right now, and we are viewing genes under carbohydrate metabolism, And then, as you can see here, we have genes, the entire genes under carbohydrate metabolism, and then we have it to be 92. So it means the 41 we have here is actually talking about let's see, we have 15 results of carbohydrates. So it's still talking about certain major the classes of genes, all right, that are assigned to carbohydrate metabolism. But once we go to this particular gene for carbohydrate metabolism, we are exploring individual, all right, individual genes of each category of carbohydrate metabolism under it, all right. As you can see here, there's so many of them. Just like we click for the metabolism, glycolysis metabolism, we have a lot of genes there. So that's it. Okay, so you could narrow down your research or gene of interest from this keyboard, I mean this search space also. All right, could do that. And then what else? So that's it. Okay, that's it. All right, so all right, so we want to see how to visualize the genome. All right, the entire genome, the visualization of genome functional features and genus software. Meanwhile, don't forget, we are still going to do a research, all right, on this. And at the research, you have, a practical understanding, a practical knowledge on how you can interpret this evaluated report, how you can interpret the evaluated report into probably a research publication, an article, or something that makes sense. All right. So we're going to be making sense out of this genome evaluation. All right. So let's take it gradually. So before we download, before we download to visualize on Genome software, like I said, we have the Contig.Fatsa, we have EMBL, we have DNA features. So let's download this. Protein, let's also download that. Features.txt, let's also download that. Gene bank, right? Gene bank contains contigs plus the genes, the entire genes in them, and the protein features, things like that. But here we have merge.gb. The merge.gb is the merge file of the genome sequence, all right? The whole genome sequence of the organism, and then we have the gene bank. So let's download this instead. Okay, so these are some of the things we need right now. And let's go to the folder where we downloaded this from. All right, so this is it. So here is the docs.txt file. So let's view this featured DNA. So this is all the future, the CDS that we have. All right, all the CDS that we have, the hypothetical proteins, protein functional assignments. All right, this is the DNA features. Why this is for proteins? Can you see these are the proteins? All right, these are the proteins, as you can see. Now this is proteins, features of those proteins. All right. So here we have the text of the features. All right, as you can see, hypothetical proteins down to every other one that we have. All right, every other one that we have. So, and then we have the match file, which is the gene bank. And the gene bank is what we need to visualize the sample on Genome software. So let's go to Genome software. From file. What's the name? All right, this is the name, Clar. All right. Match. Can I see? Dot match. This is what we need to upload. Is uploading. I want to visualize the genome, entire genome feature of this particular genome. All right, it's processing. All right, so we have it here. All right, so we have it here. Now, can you see all the tRNA? Let's try to click this. So here we have, can you see? Can you see? Can you see? Can you see? So, and then this is just what we can see, right? But let's go to annotation icon, annotation and tracks. So can you see the CDS? This is the total number of CDS we have. Faster record is the Contig. Can you see 791? The source is also the Contig 791. The tRNA is 7, which is what we are seeing now. All right. So when we zoom, we'll be able to see more features. Can you see? We have more features to view. We narrowed down our research from the features here. All right. Just like we narrowed down our research from the features here, we can also do the same on Genome software, but narrow down our research on here. So let's say, can you see we have belting? We have just one belting for the fungi sample that we submitted. Can you see? We have just one. But if it is any other organism like bacteria, we could have more, right? We could have more. As that. So, okay, we don't have for glucose, but then you can just search here and definitely you get something. All right. So when you download that, instead of working on, though Patrick will give you more information than this. Patrick will give you more information because when you click one, you have more information by the left here. Can you see? You have more information to view by the length, by the length, the sequence, the accession, the end, strand, the location is found on the genome, feature type, CDS, taxon, genome ID, genome name. As you can see, so view the circular form of this genome on Genome software. Let's go to sequence, circular, click this icon. Let's wait for it. Right, so here's it. Now the reason why we're viewing this way without viewing more of the annotation is because there's so many of it there, right? And sequence itself is so long. Can you see? It's really long. Let's click so, and if you try to can you see, if you try to zoom, we have more of those features. Can you see? So this is another way you can visualize. So but if you still want to do another round or another way of visualization, just go to this icon, click near view, click wrap and zoom. So you have more visualization like this. Good. To have more visualization, search. So you could do it this way also. You could remove this to have it here, and you could save this as image, save image file as this JPEG picture, and then we have like resolution. Try to increase the resolution. We have this. You can shade annotation. Then okay, it's done, and you can click OK. So we want to save. Let's save our download to be annotation. Right, so let's save. Saving. All right, completely saved. Can you see this is the annotation? Let's view it. You could view the rand form also. Can you see? So this is the annotation that we view. All right, this is the annotation that we view. So you could save this also. Save image file. Increase this to like 500. All right, so let's click annotation to saving as JPEG. Saved. Here it is. Let's view. So here is it. All right. So the last thing I'm going to have to show you now is the Genome browser. So when you go to Genome browser, you have another kind of visualization of the genome. Okay, now that should be circular view. Let's go to circular view. Should be circular view. Let's view from the okay. But this fungi does not have circular viewer. All right, so let's check from here. So also check the genome viewer. All right, can you see? So we have the chromosome, which is the Contig, CDS. We also have other genes included, which we are going to explore as we go on to the next professional videos. All right. So probably you might not find the Genome browser. All right. It's because of the level of professional course we are currently engaging. So once we move to comparative genomics, all right, we'll be able to explore much more and view the circular genome from Patrick. All right. So thank you so much. If you have question, you can ask a question. Meanwhile, in the next video course, that is on comparative genomics of NGS data. All right. We're going to be exploring more of this, how we can, do some comparative study between different genomes, and then, use the functional genomes like subsystem categories. All right. Certain genes of interest. We're going to be focusing on some of those genes of interest and do comparative analysis. So we look at how we can compare the result of one genome from another. So, and in that particular course, we also explore a project where we'll be able to, have a practical knowledge of, how we can interpret our evaluated results into a publication article. You understand? For the analysis that could emerge in the future. All right. So we're going to be doing a lot of things comparative genomics using the basic understanding you have right now about functional genomics. All right. Using this, and we explore more things like the circular view, how we can do a comparative analysis of different genomes in the circular view, right? So using the circular view, which is pangenomics, and also we'll be exploring phylogenetics. What are some of the ontologous genes? How can we find ontologous genes, right? Common genes, in different samples. How can we find them here on, on Patrick? How can we download it? What do we need to do to, to get a sequence, and, so that we can do phylogenetic, analysis, like based on comparative study. All right. So including the some specialty genes like AMR, antibiotic resistant genes, shared genes, things like that. So we'll be exploring that in the next course, which is comparative genomics and of NGS data, right? Which is WGS raw reads samples, both a bit single read or pair read, right? So anticipate that in the next course. Thank you so much.